Hello and welcome back to the second video in our chapter 4 lessons. So this video we're going to be covering the accessory structures. I think your book might use the word appendages of the skin. Appendages, accessory structure, the same thing. And then I'm going to do a little bit of some clinical discussion on burns and cancer, which are the most obvious kind of or most common ailments affecting the skin. All right, so we just got done covering the structure of the actual integumentary system, the layers of the skin, the epidermis with its 4 to 5 layers of stratified squamous epithelium, and the dermis with its 2 layers of different kinds of connective tissue. And we talked about melanocytes and things in that as well. So when we are talking about accessory structures, we're going to be focusing on glands. We have sweat glands and oil glands. And then we're going to talk about nails and hair. So hair and hair follicles and nails, like fingernails and toenails. All right, so let's get started. So you have this general picture of a piece of hairy skin or thin skin, since your palms and your soles don't have hair on them. So anytime you see a picture with hair, you automatically know it's going to be a thin skin sample. So this diagram is a good one to review. So if you're studying and reviewing for the lab practicum and the lecture exam, if you can find a blank picture of this in your lab manual or on the internet or in your own book, you can kind of put maybe Post-its or taped paper over the answers and just label it over and over and over again until you can start identifying these things, be comfortable spelling the words and coming up with words on your own without looking at anything. Those are great ways to learn and remember all of the parts and pieces, or what we call the anatomy of the skin and these accessory structures. So some of the things that on this, I'll just highlight real quick stuff that's not accessory structures. So dermal papillae, which was part of our dermis, epidermis, dermis, and we didn't really talk too much about this before, but I'll mention it here, is just under your dermis, there's a layer of fat called the hypodermis or subcutaneous tissue. So that's just your subcutaneous fat layer. It's padding between your skin and your muscle or bone. And blood vessel, there's your fat tissue and blood vessels and arteries and veins. I'm trying to think. So those are all things we just covered in the structure of the skin. Some other things that I'm going to highlight that aren't necessarily going to be discussed here, but you should know about them, you'll be learning about them in lab, is the nerve innervation of your skin. So we have these guys here called Meissner's, or sometimes called tactile, get the right color. So sometimes these are called tactile receptors or tactile corpuscles. Oops. They help you feel light touch. So like a bug crawling on you, or for me, like when I feel hair on my arm, I'm like, I gotta get that hair off. If I can spell it right, corpuscle. So tactile corpuscles, free nerve endings. Oops, that's the wrong grain. There we go. Free nerve endings are usually pain, temperature, kind of more damaging type of stimuli. We also have, here's just a nerve fiber going up to feed your Meissner's corpuscle. Pacinian, also known as lamellated. Lamellated corpuscles are for deep pressure and deep touch and some vibration, because you see how deep they are. So if you're going to just do a light touch, I might be activating my tactile corpuscle or my free nerve ending. But if I'm going to push hard, then I'm going to activate my Pacinian or lamellated corpuscle because I have to push down through all my layers of skin before I can activate that. Okay. And then lastly, we have the thing that gets activated when somebody pulls on your hair, or bugs walking across your arm and tickling your hair. Not only are you picking that up in your Meissner's corpuscle, but you're also, if you're moving your hair, your follicle is going to feel that. And so there's some nerve endings there. So what that's left with is all of the structures associated with our accessory organs. So that's going to be your hair shaft, the pore of your sweat glands, oil glands, what's left? Oh, your sweat gland, hair root and hair follicle. There's your eccrine sweat gland. I forgot one. I forgot another thing. Your goosebump muscle. This guy right here, erector pili muscle. It's attached to your hair follicles, and when it contracts, your hairs go whoop, stand on end, and that gives you goosebumps. All right, so we're going to take a look at, it looks like we're going to do the glands first. So let's keep moving. All right, so here's a slightly different picture of the same type of idea, but we're going to take a look at your oil glands first. So oil glands are called sebaceous glands. They are always going to be in association with a hair follicle. So if you'll notice, here's a hair follicle here. I'm kind of highlighting it in yellow. And the sebaceous glands are often kind of hanging off the side like these saggy sacs. So here's some over here. That yellow doesn't really show up. Let's do green. Okay, so these green highlighted structures, those are sebaceous glands that are hanging off and associated with the hair follicles. They produce oil. So, you know, when you touch things, you leave fingerprints, or you put your face up against a window and you've left your face print. This is oil. It helps to condition the skin so it doesn't dry out. It helps condition your hair follicles and your hair shafts themselves. So these are your sebaceous glands associated with hair follicles. Okay, and the secretion is called sebum. So instead of just calling it oil, it's got a fancy name, sebum, which kind of gives you a clue to the sebaceous glands. The next glands are called eccrine glands. These are your sweat glands. So they are what it's secreted when you're trying to do that thermoregulation. Oh, okay. There's two types of sweat glands. So the eccrine are your sweat gland for thermoregulation. This is what they look like in the microscope. They're a whole bunch of tubes all coiled together. So when you make a slice of it, it looks like a whole bunch of coiled tubes. And so they are associated with thermoregulation, temperature kind of sweat. There's another gland that's not pictured here, but your book does mention it. They're called apocrine sweat glands, or sudoriferous is a big fancy word for sweat glands. Apocrine sweat glands are found primarily in your armpits, around your nipples, and in your groin. They're also associated with hair follicles, but they're more of the body odor type of sweat. So you're usually wearing deodorant or antiperspirant to prevent this type of sweat. It's not so much for thermoregulation. We really don't know what its function is, but its content is a little bit more protein rich and nutrient rich. And so the bacteria that live on your skin love to eat it. And when they eat that protein and stuff and lipids in your sweat and your sticky armpit sweat, that's what gives you BO. It's a bacteria action of consuming your apocrine sweat secretions. Eccrine sweat glands are all over your body, like all over. Palms, soles, all over your regular skin. Apocrines, armpits, and other places. Okay. The next structure we're going to take a look at is hair and hair follicles. So I have a slice of hairy, thin skin over here on the right, kind of stained a little bit differently than the pink and purple. It's got some more blues and kind of purples and reds. No, yellows. That was weird. So here's the boundary between the dermis, and you can see lots of adipose tissue. So this is that adipose-rich subcutaneous fat layer. Here's your papillary layer. So this is the reticular layer, here's the papillary layer, and it's really thin skin. So I'm just going to call that the epidermis. We can't even go into all the layers there. But these big purpley things are hair follicles. So there's 3 big hair follicles kind of shown here, anchored into the dermis, maybe a 4th one kind of shown here. So the hair follicles are multiple layers of connective tissue. So we can see here the dermal and epidermal sheaths. That's kind of the purple and then tan color. And then the hair itself is outlined in brown. So kind of starting here, which is the same thing here. And it's got 3 layers. The outermost layer is called the cuticle. The middle layer is called the cortex. And the very, very middle layer is called the medulla. So here's those layers here. Cuticle, cortex, and medulla. Your hair is dead. That's why we can cut it and we can pull on it and it doesn't hurt, because it's just dead residual, protein fibers, collagen fibers, keratin fibers, and dead epithelial tissue, skin. as the cell grows. Most of the growth happens right here. This is the area that's called the matrix. Okay, the growth zone or the growth matrix. So this is where it's vascular. There's blood vessels that are going to come up in here and, produce, give nutrients and oxygen and take away waste products. So these cells are all living. So these cells are right here. These are all alive. And then as they go through mitosis, they're producing more and more and more cells, and they're making the hair grow, this hair shaft grow. And as those hairs get further away from that blood supply, they start to die. But that's okay. We don't want living hair. It helps to protect us from the sun rays. It kind of pads our head a little bit. So we don't want living cells to be our hair. So the follicle is the anchor that holds the hair shaft into the skin. And when you pull out a hair, yikes, it does hurt because there's nerves that are innervating. We saw that on the other picture, all those nerve fibers that innervate that follicle. So down at the base, there's life, there's living cells, and there's nerve endings. But out here on the hair shaft, there isn't any. We can see on the drawing diagram here, we can see the sebaceous gland, right? So we talked a little bit about that. Here in the micrograph, these little sacs that are kind of hanging off the side here, those are going to be our sebaceous glands. And then here's that goosebump muscle, the erector pili, and there's actually one right there in the micrograph. All right, so that's your hair and hair follicle. All right, our last accessory structure is fingernails and toenails. So I always use this picture. It's she's a great looking lady, but she's got really long fingernails. I think she was a record holder for some point in time. But I just really like that picture. So you have your own fingernails and toenails to look at as you're going through the anatomy, and you can practice when you're labeling. You can practice labeling your own fingernails. But just like the hair, there is a nail matrix. Right down deep underneath where your nail is, these are going to be the living cells. And as they're going to go through mitosis and then replicate, they're going to push out new nail material, which will again be dead and keratinized and full of collagen fibers to give you the nice, protective covering over the tips of your fingers. So it protects the tips of your fingers and your bones so you don't damage those, because you're interacting with your world with your fingers a lot. All right, so let's take a look at some of the parts and pieces. So the major body of the nail is called the body. That's nice, it takes up most of the space. And then you have on the sides, you have what are called lateral folds. So they can just come up here, kind of fold over, hold the nail in on the sides. And then we have the cuticle, which is a little bit of a residual leftover dead cells. So we can see that here in that cross-sectional view. We can also see in this view here what's called the lunula. I don't know if you guys can see it on my thumb. Oops, let me show my camera. There you go. So I have a pretty good lunula on my thumb. It's a little pale crescent area, and this just shows the point of attachment of your nail to the underlying tissue. On the sagittal view here, we can see the nail matrix. We identified that, the nail root, which is going to be growth from that nail matrix. And then right before the cuticle, we have this proximal nail fold. So just like we had the lateral nail folds kind of coming in on the sides, we have the proximal nail fold on the back side, proximal, because it's close to the beginning of the body, holding the nail in on that towards the base of the nail. And then just the free edge is what you tap, your free edge of your nail. All right, so those are our finger, and it's going to be the same for your toes. So those are our accessory structures. We went over our two types of glands: cutaneous glands, which would be sebaceous glands for oil, sudoriferous glands for sweat, both eccrine and apocrine. And then we went over hair, follicles and hair itself, and nails. So now we're going to wrap up this session or this video with some of these problems with skin. So the first one we're going to take a look at is burns. So I'm going to show you the 3 degrees. So there's 1st degree, 2nd degree, and 3rd degree burns. And these are based on how much the damage goes down into the skin. So sometimes they're called partial thickness or full thickness burns, if you've had any experience with people getting burned. So 1st degree burns only impact the epidermis. They do not go deeper than the dermis. So if you'll notice my orange highlighter here. So here's that boundary that squiggly basement membrane. So you can see in the drawing, the damage does not go down into the dermis. So this is called a partial thickness burn. It's usually indicative of just reddening of the tissue. There's no blisters. Sunburns are a good example of a first-degree burn. All the rest of the skin structures are intact. Second-degree burns are also called partial thickness burns because they don't go all the way through the skin. Again, here's that boundary. But if we take a look at it, second-degree burns do go down into the dermis, but not all the way through the dermis, because here's that boundary between dermis and adipose tissue. So there's still some intact dermis. A lot of times with second-degree burns, it's very painful because you've damaged a lot of the tissue, and there's swelling and pain, and there's blisters. So you see blisters a lot more with second-degree burns. Okay. The last type is called third-degree burns, also known as full thickness burns. And you'll notice the damage in this case has gone all the way down to the muscle. It's past the epidermis, it's past the dermis, it's past the adipose tissue, and it's gone past the connective tissue fascia and down into the muscle. So this is our full thickness burn. So full thickness burns tend to not be as painful in the site of the third degree burn because you've just destroyed all of the receptors. There's nothing left to even pick up any pain. But very rarely will you have an isolated third degree burn. It is probably also going to be surrounded by second and first degree burns, and that's where they can be quite painful. I do have a picture of a third degree burn on the next slide. It is kind of graphic, so just a little bit of a disclaimer, a warning for that. So fast forward through it if you don't want to look at it too much. So this individual, most of the third degree burns are going to appear white or black, kind of ashy, where most of this is probably going to be like our second degree, and this is our third degree up here, kind of down here. Okay, so burns are, the problem with burns is the people are going to be dehydrated if they are burned over large portions of their body. I'll fast forward. So I won't talk about cancers, but I want to have that picture kind of still stuck up there. So when they've lost that protective covering of their skin, they're open to dehydration. They're open to infection. So dehydration, maintaining your electrolyte balance is like the #1 treatment that you need to be dealing with. And then you can focus on pain management and, well, pain management doesn't really impact anything, but dehydration, fighting dehydration, electrolyte imbalance, and then take a look at infection. So dehydration is more of an important priority before infection control. Okay, on to skin cancer. So skin cancer is, well, cancer in general, because I don't know if everybody knows what cancer is. Since you guys just got done learning about mitosis back in chapter 3, cancer is uncontrolled cell growth. So all the prophase, metaphase, anaphase, telophase, where we go from 1 cell to 2 in a normal growth and repair and maintenance cycle, that's great. In cancer cells, there's mutations in the control mechanisms that say, oh, instead of 1 cell going to 2, it's lost that stop mode. And so it's just 2, 4, 8, 16, 32. So you get this huge mass, and that's what we call a tumor. So skin cancer is where those stratum basal or stratum spinosum cells or the melanocytes have some kind of DNA mutation, usually caused by exposure to UV light, and they just replicate until you get this cancer, sometimes called a carcinoma. All right, so there's 3 types of cancers we're going to talk about. There's 2 carcinomas and 1 melanoma. So the first one is called basal cell carcinoma, and just like the name implies, these are cancers of the basal, stratum basal. So it's pretty common, but they're also not the 5-year survival rate, which is usually how cancers are compared, is about 95%. So if you catch your basal cell carcinoma, treat it, remove it, you have a really good chance of not dying from this particular type of skin cancer. The next one is called squamous cell carcinoma, which is more of the mutation happening in your stratum spinosum, a little bit higher up in your epidermis. Again, this is, if you catch it early, it has a very good, about 95% 5-year survival rate. So basal cell and squamous cell carcinoma, pretty common, but also pretty treatable and not as life-threatening. The last type is called malignant melanoma, and this is a cancer of your melanocytes. So those cells that produce the pigment to protect your skin, they are what are becoming cancerous. And so they replicate, and so you get these dark kind of moles and patches and things. And this is malignant. The word malignant means it's going to spread. So malignant melanoma, these cancer cells spread more easily in your body than some of the other two that we talked about. And the 5-year survival rate for this one goes way down to like, if you catch it early, before it metastasizes, it's like 99, 95, 99%. So much like the other two. However, if malignant melanoma has metastasized to other locations in your body, that 5-year survival rate goes all the way down to 14%. So this is a very serious condition. That's why it's important to check your body, look for any changes in your moles or freckles or developing of new markers. And we're going to take a look at some preventative things to look at on the next slide so you can take care of yourself, your skin, and your loved ones around you. So these are called the ABCDEs of cancer. So A looks for asymmetry. So if you had a cancerous mole or something, symmetry, a freckle or a mole that's symmetrical looks like that. Asymmetrical might look like that. It doesn't have symmetry, okay? The border, I guess I can keep these up here. So B for border is if the border is nice and smooth, then that's going to be normal. If it's kind of irregular, kind of like my drawing here, I'm adding an irregular border. So that is also a sign that it might be cancerous. Color. So if it is nice and evenly colored, not blotchy or white or dark patches, then you're probably good. But if it's splotchy with different colors, maybe some dark aspects to it, okay, that might be something to get looked at. For diameter, we're looking anything bigger than the size of a pencil eraser. So I'm guessing, I don't know, I guess it depends on the size of screen that you're looking at it. Anything larger than that is something that you should get checked out. Small freckles usually aren't going to be a cancerous issue. But the most important one is E. E for evolution, basically just looking at change. If you have a large mole, larger than the diameter of a pencil eraser, but it doesn't change very much, it's got, its particular characteristics, just keep an eye on it. If you're looking for anything that's changing over time, and that's going to be a clue you might need to get something checked out. Okay, so that wraps up our second video. So we covered all of our accessory structures, had a conversation about burns and skin cancer. All right, so that finishes chapter 4. The next one will be jumping into the skeletal system with chapter 5. See you next time.